Hi, my name is Nicole Leidig, and this is a presentation on the amygdalo-parabrachial pathway that regulates pain perception and chronic pain. So, as an introduction, chronic pain affects over 116 million people, and many attempts to treat chronic pain often overlook cognitive, attentional, and emotional aspects. And I believe that finding an alternative solution could mean a decrease to opioid addiction and medicational dependency. Some background on the parabrachial complex is that it is a key structure in the perception of pain. It plays an important role in other functions such as appetite, sleep, and cardiovascular functions. And for this experiment, CCI-ION, which is chronic pain after chronic construction injury of the intraorbital nerve, is associated with amplified activity of PB neurons. So for our subjects, we had a total of 39 mice and 68 rats, each with 3 separate subspecies. Next, we have the anatomical, immunohistochemical, and neurochemical technique. So with the different types of techniques that they used, they ranged from tissue preparation to viral tracing, then identification of inhibitory neurons, then retrograde identification of CEA PB neurons using RNA-scope, ISH, and immunochemical ways. Next, we have the surgical procedure. So first, for the induction of chronic orofacial pain, animals were anesthetized and an intraoral incision was made along the roof of their mouth to free the intraorbital nerve. Then after surgery, animals were monitored for 5 to 7 days as they recovered. Next, we have the viral construction, where animals were placed under anesthesia and a small craniotomy was performed over targeted locations. These viral constructs were then inserted and the pipettes were left in place for 10 minutes before being slowly retracted. So finally, we have the cannular implants. These are used to optogenically manipulate the parabrachial complex. Next, we have the different manipulations of the parabrachial complex. So first up, we have inhibition of the parabrachial complex, where a single guided cannula was implanted right above the parabrachial complex. 2 weeks after recovery, a baseline was tested. These animals then went through CCI-ION surgery, and 2 months later, the mechanical sensitivity, along with going through the Grimace scale, were tested. So second, we have the disinhibition of the parabrachial complex, where a single cannula was inserted into the right PB. 2 weeks after recovery, a baseline was tested for, and then animals were randomly assigned into one of 2 groups, either receiving gabazine or receiving saline. Then these withdrawal thresholds were tested and compared to the baseline. Finally, for our third manipulation, optogenic activation of the CEA PB terminal, where rats were injected with viral constructs and implanted with fiber optic cannulas. They were covered for 9 weeks before baseline was tested, and then hind paw stimulation was collected. Next is the behavioral assessments of pain and hyperglycia. So our first method is mechanical sensitivity, which is used to assess tactile sensitivity for both the bilateral buccal region and the hind paw plantar region. For both of these, animals were placed on a metal grid, and von Frey filaments of different forces were placed on the 2 regions. Withdrawal responses were characterized from there. Next, we have dynamic allodynia, which is used to assess dynamic mechanical sensitivity. This is very similar to the first method, except it is a brush stroke instead of a mechanical probe, and these responses are characterized as lifting, licking, or a flicking behavior. And finally, we have the Grimace scale, which is used to assess pain levels based on face and body positions, looking at the nose, cheek, whisker, orbital, and ear positions. So on to the results. So first up is how the CCI-ION results in signs of pain. As you can see, the average Grimace scale for the CCI-ION mice is 1, but for the control group mice, it is 0.5. Then for the mechanical sensitivity, the average is 1.2 for the CCI-ION mice, and the control group mice are 2.8. These both show that the CCI-ION mice are in pain. So next we have the parabrachial neuron activity, which is amplified after CCI-ION, and we learned that in these mice, they are 4 times likely to show after discharges, with a 77% tactical response neurons in CCI-ION mice and a 20% in sham mice. So the parabrachial neuron activity is amplified after the CCI-ION in responses to the magnitude changes for facial and hindpaw stimulations, as well as the spontaneous firing rate of PB neuron differences. The inhibitory inputs to the parabrachial complex are reduced after CCI-ION. The changes in frequency of MIPSC and the changes in amplitude of MIPSC. So to find the source of inhibition, the PB is reported to only have a small number of inhibitory neurons. To do this, we took a picture of the coronal section of the mice, and as you can see, there are very few bright white spots in the center of the picture compared to the outside. So continuing from the last slide, there's a small number of PB neurons that express VGAT, and as you can see in the bottom right picture, these white arrows are showing just where those VGAT neurons are located. So the CCI pain results in suppressed CEA-PB inputs per the PPR recordings in mice and the amplitudes of the first IPSC in mice. In this slide, we are looking at the images used to identify CEA-PB neurons. The FG positive neurons appear white, the somatin neurons appear green, CRH or GYN neurons appear red, and the blue color is from DAPI stain. Next is the casualty. Next is the inhibition of the parabrachial complex using the 3 methods of assessment that we learned about earlier. First, we have the facial mechanical sensitivity threshold. This is the hind paw mechanical sensitivity thresholds. For the last 3 slides, the baseline medians were taken, CCI-ION surgery was performed, and then either saline or muscimol was given to see how they would affect the scale scores. Similar to the last slides, but this is with disinhibition of the parabrachial complex, they were either given saline or gabazine. Next, we have mechanical withdrawal. This is dealing with activating the CEA-PB terminals in both Windstar rats and CRH-CRE mice through the right and left paw thresholds. Next, we have dynamic allodynia. Similar to the previous slide, both in Windstar and CRH-CRE mice, both the left and right hind paws were tested, only this time they were using the dynamic allodynia test instead of the mechanical sensitivity test. For these next 4 slides, they are all to do with formalin-induced pain. The first one is optogenic stimulation in WT rats. The next is optogenic activation in CRH-CRE rats. The next is with dynamic allodynia thresholds of Windstar rats. And finally, dynamic allodynia thresholds of CRH-CRE rats. So finally, we have the Grimace scale. This was done before and after formalin injections and tests the baseline median, light stimulation median, acute pain median, and optic stimulation median. And finally, discussion. So for the source of parabrachial inhibition, it was determined that a small portion of neurons in the parabrachial pathway were GABAergic neurons, which are inhibitory neurons. So for amplified activity of the parabrachial neurons, the experiment has concluded that a chronic neuropathical pain of the intraorbital nerve results in lasting amplification of the activity of the LPB neurons. So what we learned from the CEA-PB pathways is that it is a reciprocating pathway that balances itself out, with the PB-CEA pathway being an excitatory one and the CEA-PB pathway being an inhibitory one. We now know that the GABAergic neurons in the CEA pathway contain 3 different neurons. There's a large populations of neurons expressing DYN within the CEA, however, we do not know how that regulates PB neural activity. And finally, some things that could be done to further change or enhance this experiment would be to use other subjects or use the same species in all trials and tests to test the role that MPB has in comparison to LPB and do long-lasting testing. Thank you.